The stage is yours.
Yeah. So, okay, today I just wanted a bit to present how we use Daniel radio, also known as zebrafish, to understand and dissect neural circuit in my research career and in our lab. So, again, we're mostly interested in neural circuits, and just basics is that neurons communicate electrically in the brain, and there is this complex neural code that is used to communicate between areas and inside the areas of the brain. And there are many, we saw today many probing techniques to understand this activity. And each one has its trade-offs, so depending on what you want to analyze and understand, you use different techniques. So actually, we're interested in reaching whole brain and single cell resolution to study circuits really at the neural code level and how the brain processes information. So how do we get there? We use the two different approaches. For once, we just use two-photon microscopy, which you might know, but basically is similar to fluorescence microscopy, but also you get rid of the pinhole in a confocal by just using two-photon excitation. It's nonlinear property of only reaching the energy required for excitation in the focal point. We transform it in a volumetric approach by using a tunable lens to change the incoming light angle inside the objective, so we can shift the focus up and down during imaging, so we can move our focal point in 3D. And finally, we image GCAM, which is a genetically encoded calcium reporter. And as you might know, calcium is very correlated with neural activity because each time an electrical pulse is produced, you usually have a release and an increase of intracellular calcium in the cell. And GCAMP is just a fluorescent protein that only becomes fluorescent when it binds calcium. So now you will say, but this is not enough to get whole brain cellular resolution in mice. The final trick is that we change the model animal and we switch to zebrafish, which is quite a simpler organism. And more interestingly, it's not transparent in adult, but when it's a larvae at 6 days and even up to a month, they're basically completely transparent. So we don't really need to do any big effort of penetrating the skull or anything else. And also their whole brain is basically 1 millimeter long, more or less, so you can fit comfortably under a standard objective. So pretty much we just shift the problem and we go for a simpler approach. But thanks to this, the final trick is also binding the GCAMP also to a nuclear localized signal, an histone, so it marks only a single cell each one, and this is important in zebrafish because the brain is tightly packed. So, if you would look at an image of cytosolic GCAMP, you will tell maybe that zebrafish doesn't have astrocytes because they're so close together that you cannot tell cells apart easily. But by binding it to the nuclei, where calcium still penetrates during activity, you basically kind of get a point for each cell. So just to give you an actual example, this is the kind of volumetric image you can get by tiling the single planes on a single image. And you can see the little cells are little dots. The contrast is not the best, but I think you can see them. And after this, we can also take a bit we just do the normal pre-processing, motion correction, and similar, but also we can take another approach that there is standard brain for zebrafish. So we can align our images to the standard brain, find the cells, and basically map each cells to each brain area, and we can start to do some analysis across the brain. And basically, we have this kind of synthetic fish at the end, synthetic brain activity, which is both spatially and temporally resolved. Again, the contrast is not great. But we can analyze this. So for one application, we actually investigated early neural plasticity. So the idea is that basically if you rear an animal in absence of stimuli, so in this case in pitch darkness, especially in mammals, it's pretty famous that there is a critical period where if you remove stimuli during that period, neural circuits don't really form. So we wanted to investigate this and also investigate if a modulator of plasticity, which is psilocin, the active form of psilocybin, actually can modulate this plasticity during development. What's interesting is that in zebrafish, there is actually no precise critical period. So it's more or less a more continuous affair, and also is much more genetically encoded, so neural circuits tend to form even in absence of stimuli. But we wanted to see if there was some subtle effect. So basically, we have 5 different points, both in time and in conditions. So we have the controls are always in red or purple if they're all there, and the treated fish are in this greenish color, and then you have 2 darker forms, depending if they receive psilocybin or not. So the idea is that first we wanted to see if there was any effect using behavior, because at the end, the only real read that you have of the fish is the behavior. So we see that if we let them hunt prey and collect, because they are already able to hunt prey at 6 days, we see that there is a big difference between the normal fish and the dark-reared fish at 6 days. What's interesting is this difference, even after you let them grow again in the light, even the ones that were reared in the dark, they stay pretty bad at catching prey. So we're getting actually the effect that we were hoping of disrupting the formation of neural circuits. And finally, if we add actually the psilocybin, we have a dose-dependent recovery of this effect. So we basically have our proof that we have this modulation. So we want to study it in the brain. Then first we went for the classic that also is visible in mammals of the neuroretinotopy. So basically in visual areas, you have a mapping of the different part of the retina in the retinotopic areas. And this also happened in the zebrafish. This is a flat example with 2 points, one rostral, one cutter. You see appearing different, activity correlated with one stimulus is spatially segregated. What's interesting is that when we actually went to give many stimuli to those dark-reared fish and to check if they were different from the light-reared fish normal, we actually saw no difference. This is just an example in 3D, but basically, for all parameters we could check, we never saw a difference. So we were a bit curious because, again, we had this difference in behavior, but we didn't see the classic disruption in the formation of neuron of retinotopy. So looking a bit in the literature, we saw that when fish were dark-reared, people saw some network effect at the level of spontaneous activity. So in our same experiment, we went to basically group neural activity in, I call them cluster, not really ensembles, because we used a different approach to cluster them together, but basically those are the traces of group of neurons that fire similarly. And we found a certain number of clusters that, and some of them, of course, correlated with the presentation of certain stimuli. I marked some of them here. And those are the spatial distribution in the tectum, which is the main retinotopic area of the zebrafish, which is the analogous of the superior colliculum, because the telencephalon is not yet developed completely. What's interesting is, of course, the number of clusters, which, of course, kind of matches the retinotopy because we're giving spatially segregated stimulus, is not affected by our treatment. But what's interesting is the number of neurons that recruited show quite a strong variation between the groups. What's interesting is it doesn't really match the shape of the behavior. We have this weird V-shaped effect that we explain by thinking about development and synaptic and neuron pruning, because initially you have this effect where the dark-reared fish have more cells recruited in each cluster, but then this effect kind of reverses later. So it's like the dark-reared fish are following the same trajectory that the normally reared fish are following, just delayed. But again, this doesn't really explain our behavior because it has a completely different evolution in time. But what's interesting is that in zebrafish, we can study again the whole brain. We didn't see effect on the cluster we could find of cells in other areas. But what we wondered is, like, is communication between the recipient areas and the actual motor areas that produce the behavior different? Because again, we know that there is some tectobulbar tract, so they come from the optic tectum to the hindbrain, and basically are the ones that are basically, if you actually cut these tracts, you stop hunting behavior from developing. So we went to see, okay, we have certain cluster in each fish in the visual areas that we know are partially affected, and some clusters that are not affected in the hindbrain. What if we check the correlation between cluster in each one of these tectum towards each one of those division of the hindbrain? And what's interesting is that, again, we see at this effect at 6 days, where we have this trend of always the normally reared to have a stronger correlation with the hindbrain. And when we actually switch to also 9 days, we start to see this similar effect of, the effect gets weaker, but still in some areas is very strong, where you see it maintains the same shape as we have in the behavior, but actually we also have this effect of the neuromodulator psilocybin actually shifting the direction in the same one that we expect. So this is basically the final result that's very interesting, is that this effect of disruption at the level of the tectum doesn't really change the retinotopy in zebrafish, but actually affects how the information is transferred to the motor areas, premotor areas. And now a bit more about our newer project. We actually also started to play a bit with genetic models, because again, what you can do with zebrafish is that you can generate genetic models very quickly, because they have a life cycle that's way faster than a mice. So you can take, again, advantage of this. And I'm actually focusing on FMR1, which again we generated in collaboration with other collaborators in our lab. And I mean, FMR1 is the gene that's involved in the Fragile X syndrome, so it causes problem in development that leads to changes in learning, memory, and social interaction. What's interesting is this last one, because we started to do some experiment again about social interaction in zebrafish, where we measure pretty much this interaction radius by putting two fish together and observing how pretty much an heat map on how close they get to each other. And we see that's different if compared to when the fish basically will be swimming randomly. And now what we're planning to do in future in our lab is basically to take this FMR1 fish and separate them and have them basically have a virtual interaction, where instead of just putting them together in a single dish, we're going to split them. And one, however, is going to be fixed under the photon. So this is our basically ongoing experiment right now. And again, it's one cool thing you can easily do with zebrafish is putting them in virtual environment, because they're very visually driven animals. And again, you can put them under tissue photon without any manipulation while giving them visual stimuli. So this is what we're working right now in our lab with Marco Dalmaschio, the previous project, and Nicola, which is also doing the genetic modification. Claudio Lodovichi was instead the PI that followed the previous project about the plasticity, and also I thank the other people in the lab, and especially Marco Salamanca, which is my collaborator for a long time. So yeah, I don't know what was the time, because I wasn't looking too much, but yeah, it's 13 minutes. Okay. So.
Thanks.